welcome to the lab. We're focused on early detection of cancers of the esophagus and stomach. And the reason that we're really interested in these cancers is because, if you like, they're a paradigm for understanding how cancers progress, how cancers start from the very, very earliest stage, from a normal cell through to a precancerous cell, which then over a period actually of years can develop to a full-blown cancer. And the esophagus, which is very accessible by endoscopy, for example, to take biopsies, is an example of a cancer which occurs very slowly. Patients are predisposed through damage and inflammation, through acid and bile, and we actually monitor patients over time so that we can really study in exquisite detail what those changes are as you move towards a cancer. And I'm really fortunate to be working in a very interdisciplinary team where we have a very translational approach to the work that we do. So the team has both clinicians, basic scientists, computational biologists, all working together on this problem. So what we've done is to take advantage of sequencing techniques and new model systems to try and really understand those steps in the evolution towards cancer. And then what we're very motivated to do is actually apply that knowledge to the clinic. And we've taken some very practical approaches to actually developing new diagnostic tests. Let me show you just one of those which we've progressed furthest with, which is an example of how you can take the knowledge in the laboratory and come up with something to apply to patients. So at the moment, to diagnose an esophageal cancer, you do that through an endoscope. An endoscopy means going to the hospital, having a reasonably invasive procedure. It's quite straightforward, but still you're going to have to take the day off work. It's quite expensive. And you take tiny biopsies, which only give you a small representation of the affected part of the esophagus. What we've developed is a very simple test, a little pill on a string from the patient point of view. When we get the sample to the lab, we can use our knowledge of the molecular changes to understand exactly where that patient is in the pathway towards cancer. So what happens in this test is the patient swallows this little pill on a string, the capsule goes down towards the stomach, and over a period of a few minutes it will dissolve and out pops a sponge. This spherical sponge is then pulled back up by the nurse. It comes out very quickly in just a few seconds. And on this sponge, you collect a very rich cell sample of over 1 million cells. And then you can use that to do molecular tests. We just send this specimen in a pot to the laboratory for those molecular tests, which we design to tell us whether the patient has the precancerous condition Barrett's and how cancerous the cells are becoming. So that's just one example of the type of approach we can take, and we take this all the way through to clinical trials. So this test is now being trialled in a patient study of 9,000 patients in the UK, a trial called BEST3. So in summary, we have a really translational, practical approach to understanding cancer better and diagnosing it early, particularly focusing on cancers of the esophagus, and it's a real teamwork effort.